Well, thanks very much for this opportunity to be here. And I'd like to warm us up to some of those real on-the-ground questions and problems that we have in the environmental health sciences community. Talk a little bit about how we're applying AI currently to those problems. But I want to focus mainly on what are the problems and how can those of you who do AI and develop those two-year-old kind of applications so we can really make progress in some of these major challenges and questions that we have in environmental health sciences. So let me just start off by telling you a little bit about NIEHS, since I understand that many of you are outside the environmental health sciences community. So NIEHS is one of the 27 institutes or centers that comprise the National Institutes of Health. So we are unlike the 26 other centers or institutes that are based primarily in the Washington, D.C. or Baltimore area. We're located in Research Triangle Park, North Carolina. And there's a good political reason for that and a good science reason for that. If you're interested, ask me at the break. So like the other 26 other ICs at the NIH, we have an intramural research program, and we have an extramural division where we take the bulk of our budget, we package it up and send it off to many of you in the form of grants and contracts. But additionally, we have a second intramural research program, and actually Nicole is part of that program. It's called the National Toxicology Program. It's a testing program where we apply our knowledge of how human biology works, and we want to better understand how environmental exposures and environmental agents interfere with that human biology. So just a quick comment about funding. How much money do you have? We're not the biggest IC and we're not the smallest IC. Our annual budget is about $850 million. We take some of that, actually package it up and send it to the National Academy for workshops like this. But again, the vast majority of this money is actually sent out to academic organizations or other institutions that conduct environmental health sciences research. So what differentiates NIEHS from the 26 other ICs is our mission and our vision. And you can see the mission on the top here. It's really about how the environmental health sciences, how the environment affects people's lives. But most importantly, I want to focus on the vision. And the NIEHS is really about providing global leadership for innovative research that improves public health by preventing disease and disability. And I want to focus on the prevention part because our institute director, Linda Birnbaum, really thinks of us as the Prevention Institute. Unlike many of the other ICs at the NIH that really focus on how do we cure cancer or how do we cure kidney disease, the NIEHS is really all about how do we prevent it from happening in the first place. So if you want to know what we do, I encourage you to take a look at our strategic plan. And we very much tie the grants and the contracts that we award to our strategic plan. So if you're in good alignment, you're likely to get funded. So take a look at the plan. And I'm not going to go through great details here. There are three different themes in our plan. The first theme is advancing environmental health science. And this is really about doing fundamental research to better understand human biology and to understand how environmental exposures interface or interfere with that human biology. The second is translating data to knowledge to action. And it's really taking the fundamental things that we learn through our research and actually putting it to work on behalf of the public health community. And the third is about enhancing EHS through stewardship and support, which means paying attention to our human resources, our non-human resources, to make sure that we have the fundamentals in place to enable our work to be done. So integral to all three different themes are elements of AI, computational science, and data science. So what I thought I'd do is give you a few vignettes, a few specific examples of how we're applying AI to environmental health sciences, or specifically these projects at the Institute. But most importantly, really kind of set us up, and I hope we'll give you some elements of thought of how can we apply new and innovative approaches to AI to really solve major problems in environmental health sciences. So let me start with what we call the Tox21, or the preventive or predictive toxicology. This is really at the core mission of NIEHS, which is to establish the toxicology associated with different environmental agents. So at a workshop here at the National Academy in 2007, we actually faced kind of the reality. We had then many different technologies and approaches for evaluating toxicity. Most of them were very expensive, they were low throughput, took a lot of time, and the net effect is that we knew quite a bit about a few different chemical agents. But the sobering reality is that there are thousands, if not tens of thousands, of things in the environment that are produced by different industries, and we know very little about the toxicology. Recently, for example, there was a Gen X spill in eastern North Carolina. So the question is, what do we know about Gen X and these perfluorinated chemicals? The problem is we know very little about them. So we have to solve that. And part of the solution was to essentially, and as a result of this workshop, there was a report, a National Academy report, which really proposed to shift primarily from the in vivo, low throughput, and not very insightful animal studies to in vitro assays. And the thought is to virtually, in the future, to virtually move to all routine toxicology testing conducted in vitro using the high throughput robotic assisted methodologies that are used, for example, in the pharmaceutical industry, very effectively in the pharmaceutical industry. So we actually embraced that report. So we developed a memorandum of understanding with other agencies, and the idea ultimately was to develop a whole set of different tools that can interrogate elements of biology that we know something about. What are the pathways and what are the networks that drive human biology? Can we set up in vitro molecular or cell-based assays to interrogate these systems and learn something about whether a specific environmental agent or a chemical is interfering with that reaction? So here's just an example. I don't expect you to read all of these, but there are many different molecular cell-based assays that are currently available in the upper left-hand corner. We can actually look at apoptosis. We actually have assays for nuclear receptors. So the idea is to employ these different assays here with different chemical agents using high-throughput robotics-based platforms, and then ultimately come to some decision on whether or not a given chemical entity is likely to be deleterious to your human health. So this is a slide that I borrowed from Nicole, and she'll be showing you this slide again this afternoon. And I think this represents the big challenge. So here we are in the upper right-hand corner with these high-throughput screening. We generate huge amounts of data with different chemicals, lots of different molecular and cell-based assays. And we have to now factor into that, the RD kit and PhysChem databases. We have omics technologies with complete transcriptomes. We have metabolomes. We have genome sequences that we have to toss into the big data here. And plus, we want to take the legacy data that we've created. So I think the huge challenge going forward is how do we take all of these different data sets and somehow plug it into maybe that good old, we'll give it a try this afternoon. Let's put Penn AI to the test, and maybe by 4 o'clock this afternoon, we'll have everything that we need to know about various different chemicals. So it's a huge challenge, and again, Nicole is going to describe the Tox21 and predictive toxicology programs in much more detail later on in the meeting. So let me move to a different project now. And we call this the exposome. This is probably one of the newest projects, and I know it's something that Gary Miller knows a lot about. And the exposome is really defined in the upper left-hand corner. It's really an untargeted, hypothesis-free assessment of the totality of environmental exposure. So what is this all about? Well, we've come to recognize that we've been very effective in looking at, well, is benzene bad for you? Is Gen X bad for you? Is lead bad for you? But the reality is that if we want to understand how the environment impacts human health, we have to factor all of these things into the equation. Because it's not just, say, the benzene that you were exposed to when you were filling up your car with gasoline this morning, but you were breathing in PM 2.5 behind the bus on the way to the center here. So the exposome project is really about trying to bring all of the elements shown here, the radiation, lifestyle elements, infections, drugs, diet, pollution, different chemical exposures, and then factoring into this the fact that windows of exposure is extremely relevant. It's not just the immediate past exposure that can lead to potentially deleterious health effects. I'll show in just a few slides that the exposures that you had when your mother was pregnant with you may actually emerge later in life and give rise to a phenotype associated with those exposures. So we have to factor windows of exposure into this. And again, this whole issue of mixtures, mixtures, real-world mixtures involving chemicals and non-chemicals absolutely have to be integral to better understanding the exposome. And then the last element, too, is the microbiome. For many years, we just ignored the fact that we have this synergistic relationship with tens of thousands of different microbial species in different parts of our body. And this has become extremely important to better understand how the environment impacts our health. So this is a huge data challenge. So I don't know, Penn IEI, are you up for the challenge here? We'll toss in all these different variables, and ultimately, we can hopefully understand truly how does the environment impact human health. So this is a huge data management and computational challenge, and I think, quite frankly, that the exposome is really an AI challenge. How do we collect the data? So we're not exactly sure. Many of the grants that we're supporting now in the area of the exposome are really all about trying to figure out how do we collect data around diet, for example, and have the type of data that we can actually use for ultimately the calculations to establish whether or not a given exposure is bad for you. So let me move into the third area, which is about individual susceptibility. So the reality is that if you look across the room and look across the internet, not only do we look different, but we actually metabolically are very different, and we respond to the environment in a different way. So what might be bad for you may not be necessarily bad for me. So if we want to establish hazards and risks associated with the environment, we have to know something about the host. And so several years ago, we joined with several of the other institutes or centers at the NIH in something we called the G by E, or the Genes by Environment Project. So on the left, we looked at diet, physical activity, environmental exposures, psychosocial stress, and addictive substances. Then we worked with NHGRI to sequence genomes and to see could we correlate any sequence variations in genes with differential responses to the environment. So I think a great illustration of what does this look like is shown on this slide. And this is where Jeff French and many of his colleagues in the National Toxicology Program looked at different inbred strains of mice, shown here on the left. And they looked at several different parameters as it relates to benzene toxicity. This is, in this particular case, it's about benzene clearance, but you can see that depending upon the inbred strain of mouse, it behaves very differently. It responds to benzene in very different ways. So one thing that this tells us is that for any given inbred strain, and in the case of the NTP, we actually use, it's an F1 between two different inbred strains, but it's a defined genetic background. And so we come to learn that any given inbred strain doesn't necessarily reflect the genetic heterogeneity that exists within the population in this room or within the human population. So what we really need to be doing is thinking about moving away from a single genetic background to developing genetically diverse mouse populations or rat populations or fly populations or C. elegans populations, because that's more likely to give us the answers that are reflective of a genetically and physiologically diverse human population. Now, fortunately for us, the mouse genetics community, I know the rat genetics community have done the same thing, and the fly genetics community have done the same things. And the idea is to create these genetically diverse populations of organisms. So on the left here, and I'm not going to go into the details on this. If you're interested, ask me at the break. But there are a couple of different resources. One of them is called the Collaborative Cross, and the other is called the Diversity Outcross. And the idea was to take 8 different inbred strains shown in different colors in the upper left and to systematically intercross them to create ultimately what we call the mixed up mice. Mice that essentially have haplotype blocks that are derived from the 8 different founder strains. You have the Collaborative Cross, which is a set of recombinant inbred strains, and the Diversity Outcross, which is a population of animals that have these different size haplotype blocks. And the question that we asked is, in the MTP program, we actually challenged the community several years ago, does it actually work? Does it give us data that better reflects what you're likely to see in the human population? So Jeff and Gary Churchill at the Jackson Laboratory and many other colleagues in the mouse genetics community actually decided, well, let's give benzene a try. So it turns out that in benzene, in the human population, we know that different people respond very differently to benzene. So if we did an experiment with just C57 Black 6J, would that data actually reflect what happens in the human population? The answer is no. But what about if we use this diversity outcross? So they actually set up an experiment, a 28-day inhalation study, and I want to focus on the dose of 100 parts per million all the way to the right. And you'll see that there are different dots, and these are the responses of individual mice in that population to that same dose of benzene. So different mice respond differently, which is exactly what you'd see in the human population. So this is a huge step forward in terms of modeling what you're likely to see in the human population in terms of establishing risk. But the next question, and this is the real challenge for AI, is actually can we find the genes in these different mice, and can we identify the variants of those genes that are responsible for this differential toxicity? Because what that does, it really gives us insights into the molecular mechanisms of toxicity, and that's ultimately where we want to go. Well, the answer is that, and again, for reasons that I don't have time to go into, these mice were generated in such a way that you can very easily go in with what they now call the Megamuga chip, and you can very quickly establish essentially the structure of the genome of these individual mice that were responding to benzene or at different environmental exposures. And you can toss this together and you can do some analysis. And on the upper part, you can see that there's a big peat on chromosome 10. And this identifies a gene, a genetic variant mainly in the Castanea strain that confers resistance to the benzene exposure. So if you toss in some additional AI and some bioinformatics, you can ultimately identify a region on chromosome 10 that has a number of genes that are good candidates as being responsible for this differential susceptibility. So that was a huge step in the right direction. So we now have a model system that is actually better predictive of what you see in humans, and it's actually giving us some information about the gene variants in different genetic backgrounds that are likely to be contributing to differential toxicity. But the question is, can you actually move this into a cell-based system? So one of our grantees, Ted Choi, is the chief science officer at his own company. It's predictive biology. It's in the greater San Diego area. And what Ted basically decided to do was to take a cohort of those diversity outcross mice, and he created embryonic stem cells from them. So if you start with, say, the diversity of this population of diversity outcross mice on the left, and on the right, he had essentially a population. So different wells of a microtiter dish had embryonic stem cells, but each was derived from a different diversity outcross mouse with a different genetic background. And so what he asked is then, can we take these and can we actually put these through a whole battery of different in vitro tests from transcriptomics, proteomics, et cetera, and find gene variants that are responsible for differential susceptibility. And he's actually very actively involved in doing this right now. He's getting some very promising data suggesting that there are gene variants that we can identify in vitro. But the fact that he used the embryonic stem cells allows us to actually go beyond simple cell-based toxicity. And that's shown in the next slide. So say that you have a cell-based system and you're interested in knowing whether or not the chemical that you're interested in is likely to cause heart arrhythmias. So what Ted has determined is that you can take the embryonic stem cells from this population of cells derived from a population of diversity outcross mice. Can you differentiate them into beating cardiomyocytes? And you may need to help me in the back. Could we click on the middle panel? And so what he discovered is, yes, you can take these embryonic stem cells, you can differentiate them into beating cardiomyocytes. And typically, these beating cardiomyocytes will beat with a regular periodicity. Okay, but then he asked the question, could you take a drug that was actually pulled from the market because it caused arrhythmias? And can you identify the arrhythmic beating pattern in these cells? And you click on the panel all the way to the right. No, there you go. You got to activate it. Is it going? There you go. Okay, so you can see that, well, hopefully you can see that the middle panel, it was beating in a very regular way. In fact, in the lower right-hand corner is the graphical illustration of the beating pattern. In the absence of drug, you have a very regular beating pattern. With the drug, you get this very arrhythmic pattern. You remove the drug, the cells go back to the regular beating pattern. So what this does is now introduces the possibility you can actually use cell-based assays to be looking at more complex phenotypes typically associated with whole organisms. So in terms of data management, you now have to not just putting in genome sequence and the expression of genes, but looking at complex phenotypes that you have to overlay on your data sets. And actually, the last thing I'd like to do is actually highlight some of the work of our new branch chief, Ellison Motsinger, who we recruited from North Carolina State University and who trained with Jason several years ago. But she's doing some really interesting experiments. And the question is, can you apply this cell-based approach that Ted had developed to humans? So what Ellison has done, and Ellison has done lots of different things, and I can't describe all of this in the minute and a half that I'm going to give to this project. But what she's doing is using immortalized lymphoblastoid cell lines, and she has this high-throughput in vitro-based cell viability assay, and actually tests to see whether there's a specific compound, temozolomide, and actually what she found, if you plot it out, this is a Manhattan plot on chromosome 10 again, there is a very strong peak, and actually under that peak was the gene called 06-methylguanine DNA methyltransferase. So she was able to take this differential sensitivity to this drug and search the genome and actually identify a gene and a gene variant associated with that differential toxicity. So this is a huge step forward in the right direction. But I think the challenge for the AI community is that, look, we identified a gene here, and we identified a handful of genes in some of the other experiments we've done. But the reality is that our biology works, we have everything that happens genetically as a complex trait. It's not just one gene, but there are multiple different genes. So the challenge is to figure out what are the networks of different genes that are associated with this toxicity, not just the one on chromosome 10. They're going to be much weaker hits. So the question is, what data sets do you need to actually plug into the Penn AI algorithm to ultimately come up with this type of information so that we can look at your genome, my genome, and have a better understanding of what type of differential susceptibility to our environment have we inherited from our parents. But the next thing I want to focus on is the whole issue of epigenetics. So we have gene variants that we inherited from our parents, and we used to think very simplistically that the variations in our genome are responsible for any differential susceptibility to the environment. Well, it turns out, I think, as we're all beginning to realize, is that there are quite a number of different either DNA methylation or histone modifications that can influence how a gene is being expressed. And many of these are influenced by the environment. So I think a great example of the epigenetics in action actually derives from work, actually in my own laboratory over 20 years ago, where we characterized a very interesting mutation in the coat color gene called agouti. And so when you look at the slide, you see mice that have different coat colors. In fact, the mouse that's yellow, fully yellow, after about 3 months of age, it loses its capacity to control its appetite. So it becomes massively obese and develops type 2 diabetes. But if you look at the slide, you see all these different phenotypes, and you just assume from a genetics point of view that they all have different genotypes, that there may be a sequence variation in the yellow mouse, there may be a different sequence variation or two in the mice that are mottled. It turns out that these mice are all genetically identical. It's an inbred strain of mouse, and they all contain and carry the mutation that has the capacity to turn the coat color totally yellow and to induce that obesity and type 2 diabetes. So what's going on here? So we actually went on and determined that the differential expressivity of this trait and this penetrance hierarchy is attributable to epigenetic modifications. So the mutation in the case of the mouse all the way at the bottom that looks phenotypically normal, the mutation has been epigenetically silenced. So there's a methylation silencing, as well as we've determined more recently that there are histone modifications that basically turn off the mutation so it's not being expressed. And this has been used in the environmental health sciences community for the last several years. Randy Gerald first did these experiments. Dana Dolinoy has done a number of these experiments. And what they determined is that what you feed mom during pregnancy can influence the number of mice that are delivered in these different phenotypic categories, ranging from a totally yellow coat color to what we call pseudoagouti or phenotypically normal. So what mom eats can influence the methylation status of this particular gene. So that's pretty interesting, and certainly of great interest to us in the environmental health sciences community. But now, of course, we have tools that go beyond just the agouti locus. In fact, my own laboratory, just a little fresh data just in the last couple of weeks, we actually were very interested in determining whether or not rotenone, which is a pesticide, and it's one of those chemicals that we know a lot about. It turns out that it very specifically interacts with complex 1 in the mitochondria. So it tickles the mitochondria and can influence mitochondrial function. So we wanted to know whether or not during, say, preconception, gestation, and weaning, whether or not tickling the mitochondria with a little rotenone can actually influence the methylation pattern across the genome. And the first question is, does it influence the coat color? And the answer is yes, it's all the way on the left. We can actually increase the amount of expression of that yellow trait. But most importantly, very interestingly, if you look at the methylome, and so this would be all of the potential CPG sites across the genome, and just without any rotenone. So mom was just eating regular mouse chow, and we looked at the methylome at day 22, at 6 months, 12 months, and 18 months. By the way, the we here is not me as much as it is Janini Santos, a staff scientist in the lab, and Ozzy Lazoya, who's a postdoctoral fellow in the lab. So what they found, they clustered this together using these AI and computational tools. So you know that there are some genes all the way on the left. So blue means hypermethylated, and yellow is hypomethylated. So there's a whole cluster of loci that start off hypomethylated or hypermethylated, and it changes very quickly at 6 months. But all the way on the top, from 12 to 18 months, there are a whole bunch of genes that actually change remarkably in their methylation pattern, when you're pretty old. So one of the questions we asked is, how does rotenone treatment actually influence this methylation pattern? So this would be horizontally across one heat map to the other heat map. On the right is 10 parts per million of rotenone in mom's chow during a developmental exposure. And I think you can see even from the back of the room that there are some patterns that are not shifted pretty dramatically, but most notably, I think I've highlighted this, here you have some differentially methylated regions that are remarkably influenced by the rotenone treatment during development. So what we can do is actually look at the genes that are close by to these CPGs and get some sense of what are the networks and the signaling pathways that are influenced by feeding mom this pesticide that tickles the mitochondria. And we think that this is happening. The effective time is in the early pre-implantation stages of development. So the net effect here is you can see that there are methylation changes that normally occur between 12 and 18 months that are not happening because of this developmental exposure. So the last thing I want to touch on here is the area of systematic review. So this is a major area of focus in the National Toxicology Program. And the idea is to start with a question. What do we know about Gen X, for example? And can we go through and then essentially scour the literature and find out everything that's known about Gen X and then ultimately come to a report and some conclusions? So the way that we do this right now is it's very labor intensive. You get a bunch of PhD scientists or trained individuals, they read a bunch of papers, they scour through the text and catalog the information, and then ultimately develop a report that's very useful. So the question was whether or not the text mining algorithms that could be used to accelerate the process or maybe even improve the process. So and Charles Schmidt, I know, is in the audience someplace. I don't know where Charles is, but as part of the NTP, they actually put to the NISC, the text mining challenge. And so they actually provided them with a number of different data sets, and they asked the question, could the artificial intelligent text mining tools actually do a better job than what individuals do? So they went through, systematically looked at text from a number of different documents, and here shown on the red would be This is the human intervention in the blue, would be the artificial intelligence. And this is simply a measure of several different endpoints here. How well is the machine doing? Kind of the bottom line, the machine is doing okay, but I think there's more developments that need to be done. So, this is a step in the right direction, but again, there's more work that actually needs to be done. So in the closing moments here, the one thing that I'm personally passionate about, and I just want to reinforce, is that if you want to use these powerful new tools and algorithms for AI, you got to pay attention to the quality of your data. Garbage in, garbage out. And so it's data science and making sure that all those high throughput technologies that you're using to generate data and the computational technologies that are being used to mine that data, the data sets have to be available, they have to be QC'd, and you have to know that they're the types of data, and you're collecting the data that's meaningfully useful to answer the question that you ultimately want to answer in environmental health sciences. So you got to pay attention to that. And essentially, since I'm out of time, I will end. Thank you for this opportunity. Hopefully I've set the stage with a few tangible examples, but it's not exclusive to the things we're interested in. There are lots of different things that are happening. These are 4 examples. And then maybe this afternoon, again, we'll challenge Jason to charge up that program and get some answers. Thanks.